this video we will discuss about this sTRA glycan and CA19 glycan reflects non-identical subset of cancer cells. In the previous video we have understand that this sTRA and CA19-9 express independently and they are not dependent on each other. That is, they are not dependent on each other for the expressions. In this video, the researchers presented in this figure. The researchers presented some of the results that shows that in some cases sTRA and CA19 expresses on both the surface both express on the cell surface, and in some cases sTRAs and CA19 express on different subsets of the cancer cells. So for this analysis they choose this TMA5, and where in the quantification is level, that is marker levels of the tissues, shows different percentage of the CA19-9 and sTRA in different cores. That can be seen here, that for H5 we can see that CA19-9 expresses more and sTRA's expression low, and similarly this variation continues for different cores. After identifying that both of them expresses at a different level at different cores, they wanted to check some of the core for in order to see whether sTRAs and CA19 are expressed on the same site or at different site. First, in the case of panel B, we can see that this is about the more differentiated cells. Here we can see that CA19 expresses more compared to the sTRA, and this orange color indicates that it's the CA19, and the cyan color indicates that it's the sTRA expressions, and the green color shows the overlap. Here we can see that the overlaps are clearly possible, and this is the zoom in image of this white square. Here we can see that overlaps are very much compared to the non-overlap regions. Overlap regions are high compared to the non-overlap regions. That is, cyan and orange are low compared to the green. It indicates that CA19 and sTRA is both expressed on the differentiated differentiated pancreatic cancer cell lines. And when they check about the least differentiated cells, what they have find out is that CA19 expresses more compared to the sTRAs, and the zoom in image of this white square shows that is the CA19 expressions is visible, whereas sTRA's expressions are almost negligible, and neither the common expressions can be seen. And for the case of in the case of panel D, there they checked about the particular organ, and in this case we can see that the expressions of the sTRAs are seems to be more compared to the expressions of the CA19, and this is visible in the overlay, where the cyan expressions are more compared to the overlap, seems to become more than the orange signals, indicating that in this region the sTRAs are expressed more, and some of the common regions are also visible, but the independent CA19 regions are less visible in this panel D. And in the case of panel E, we can see that the expressions of the sTRAs here also is high, and here the dual expressions are very low. Here for example, the overlays on the right hand side shows that the cyans are very high compared to the green signal, and the orange signals are almost negligible. So in the case of this panel D, these regions, and panel E regions, we can see that sTRAs are dominant compared to the CA19. And when they check out this fluffy cytoplasm area, here they can see that the expressions of this CA19 are also there, and in the neighborhood of CA19 we can see the expressions of these sTRAs, and there are common regions also that expresses both CA19 and sTRAs. But the important thing is that the sTRAs are expressed on the differentiated pancreatic cells, cancer cells that has CA19. This indicates that the sTRAs could be expressed in the neighboring in the cells, in the neighboring cells that contain CA19-9 expressions, and thereafter they check it on the different cores where they have find out is that the dual expressions are high. Dual expressions are high in the case of panel E, where they can see the orange expressions are high compared to the cyan or green expressions, indicating that it's the CA19 are expressed in this panel E core, E1 core. And when they check about the panel F core, they see that the expression levels of this cyan or sTRAs are dominant than the overlap or the CA19 expression levels. All this data indicates that the CA19 and sTRAs are expressed in some cases independently and can reflect the different subset of the cancer cells. However, in some cases they overlap, and in that case they expressed a different subset of pancreatic cancer cells. So when they are uh, expressing at the same cells, they show reflects a different subset of cancer cells, and when they are expressed independently, they also reflects a different populations of the pancreatic cancer cell subset, different subset of pancreatic cancer cells populations. This is what they want to make a point out of this figure. I hope you understood the point, that is that the point that they wanted to make from these results, that sTRAs and CA19 reflects non-identical subset of cancer cells. If you need to understand more about their experiments and results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.